So what we first saw in the Moderna sample at a normal regular microscope, which uses light, is we saw various little crystal shapes and patterns on the slide. And then we did the platinum coating on it to get a closer look. And as we got a closer look, we see on top of these little crystal things, these shapes that look like chips, right? You see that square, well, that rectangle, it's got all these little dots on it arranged like a grid. So you see those little square boxes there that say spectrum 14 and spectrum 15. That's when we're telling the electron microscope to look at that little rectangle and tell us what elements are contained within those squares.
Okay.
So if we scroll down on the file, we'll see spectrum 14, and this is the X-ray diffraction spectroscopy. So as the electrons that are added to the sample are released, they have a certain energy spectrum. The most prevalent element in that little square, which was spectrum 14, is carbon. And then the next most prevalent atom is oxygen. The very first peak there, it's just kind of a general release of energy from the sample after it's been bombarded with electrons. So that one isn't assigned any element. There's certain elements that X-ray diffraction spectroscopy can't detect. And one of those elements is hydrogen. So we don't know how many hydrogens are in this sample because that particular machine can't detect hydrogen. Okay. So it looks like it's exclusively carbon and oxygen. The PT are those peaks that come up when there's platinum in the sample, right? And that's platinum we added to the sample so that it doesn't burn up when exposed to a strong electron beam. PD is palladium, and Cl, there's a little bit of chloride, which we would expect to see in anything that used to be in a salt solution. Now we'll get to the thing that looks like a little computer chip with all sorts of dots in it. And that's where we have carbon and oxygen again, platinum, palladium, and a little bit of chloride. It looks like a computer chip, but it's made of completely of carbon and oxygen and an unknown number of hydrogens. So again, this is not anything that I'm aware of, but it certainly isn't made of any silicon that all the computer chips we know of are made of. Okay. The other thing it could be is it could be like a little plate made of carbon and oxygen. The other, potential options for that is it's a rectangle of graphite or graphene. It doesn't look, nothing on the picture looks like a crystal of carbon, right? A pure carbon crystal would be something like a diamond. Other forms of carbon are carbon fiber, which is little ropes of carbon, and graphite or graphene, which can take any form from sheets of carbon to little squares of carbon. When left at room temperature, or the various temperatures that you'd have in a car for a month or more, these samples seem to organize into structures that I can't explain. The structures look like organized little chip structures, but they're not made of anything that normal chips are made of. They seem to, when they dry up, these samples dry up, they crystallize into crystal shapes. But again, it's not a salt crystal, which would be sodium and chloride, NaCl. It seems to be some kind of crystal shape made up purely of carbon and oxygen, which is very unusual. So the interesting thing, before we get to the next Moderna file, is that the X-ray spectroscopy didn't detect any nitrogen or phosphorus. So if those complex shapes, that rectangle with all the dots arranged in a grid, was the result of some kind of biologic process, like, I don't know, contamination of the sample from mold or bacteria growing in the sample, then there should be nitrogen and phosphorus there in addition to carbon and oxygen, because every living thing, whether it's a virus, plant, or animal, is made up of proteins that contain nitrogen, carbon, oxygen, and phosphorus.
So very confusing and curious. This is the next image that you shared with me. Let's get into what this looks like to you.
So when I first saw that image, I thought, oh, well, that could be anything, right? That could be a mold spore that is starting to sprout little legs, because mold spores eventually will grow into a mold or a mushroom. So, when a mold spore sprouts, it has to start growing little roots. That could be what it is, right? And that's what it looked like to me initially. And I thought to myself, well, is this mold contamination of a Moderna sample? But just as we saw before, we use the electron microscope to determine what elements are in the picture we're looking at. So there's 3 squares there. There's spectrum 20, spectrum 21, and spectrum 19. So let's scroll down and see what was included, what elements were in spectrum 19. So spectrum 19, again, we see carbon, oxygen, a little bit of calcium, a little bit of sodium, a little bit of magnesium, a little bit of aluminum, 0.7% by weight, silicon, some sulfur, and then some more calciums. So calcium, sulfur, and sodium, those would all be expected in a biologic sample. So the big question for me is, where's the nitrogen? Where's the phosphorus? Because some proteins do contain sulfur, so sulfur certainly wouldn't be unusual. Aluminum is not supposed to be in any biologic protein, but sometimes cells get confused, and instead of having calcium in a particular protein or enzyme, the cells can mistakenly incorporate aluminum instead of calcium. So that's why aluminum's probably very bad for people in the long run. It's not immediately lethal, but if you have enough aluminum in the body can get confused, and the cells will start putting aluminum into their enzymes and proteins instead of calcium like they're supposed to. So again,
so does this say then that likely there is aluminum included in this is a Moderna vaccine, potentially?
Yes. Okay. This shape, this ball with the legs growing out of it, for some reason has aluminum in it. And I can say with certainty that this isn't a mold spore or some other type of biologic contamination, because the only thing in it is carbon, oxygen, and no signs of nitrogen, no signs of phosphorus, which would indicate something biologic of origin. So this thing that's growing is non-biologic. And it was found inside a Moderna sample that was left unrefrigerated for anywhere between 1 and 2 months.
Now, how you've made the comment it's growing. How are you witnessing that this is something that is growing?
Well, I don't have 100% confirmation that this is growing as the sample evolves. We should be getting images sometime in the near future where we have an electron microscope attached to a time-lapse camera that can examine to see, how these structures change with time. But certainly I can't imagine that these ball and leg-like things are already pre-made and then frozen and contained within every single one of the Moderna vaccines. So then let's scroll down to spectrum 20, and that's another part of this ball and leg type structure. And again, we see the same thing. It's predominantly carbon and oxygen, no signs of nitrogen or phosphorus that we would expect from a biologic organism. And we see some calcium, aluminum, and a little bit more silicon in spectrum 20. And then let's scroll down again to spectrum 21.
Now, when it comes to a vaccine, would you see silicon ever for any particular reason, any kind of coating, or like what
Not that I know of. I mean, silicon can be contained in various objects like sand, has silicon atoms in it. But again, not that I'm aware of.
Okay.
And then again, the other part, spectrum 21 of that structure, is carbon, oxygen, calcium, and silicon, with a little bit of calcium. So again, there's no signs of biologic elements, nitrogen or phosphorus, in any of what we saw that looked like mold spore sprouting legs. So this was also seen in the Moderna sample, the same batch of Moderna that we were looking at before that had these chips and crystals in it. So then let's move on to the third Moderna file. Well, the only thing I can say for certainty is all these strange structures we're looking at from the Moderna sample, they're not of biologic origin. So there was no biologic contamination of these samples.
Okay.
So this next one, so this is the most zoomed out photo. This particular structure was, I think it was big enough to see on an optical microscope, although I don't have any optical microscope pictures for this particular presentation. But if we scroll down, we'll zoom in on it and get a closer look of what we're looking at. So we see a strand on top of some crystals. So if we zoom out or zoom into the particular, the folded over part of that strand, so if we scroll up again, if you look at the right-handmost part of the strand, it looks like the strand is folded over, and the folded over part of the strand appears to be sitting on top of a crystal. So then, so in the middle of that strand, there are some little things that look like dots in between the folded over parts of the strand. So when we scroll down, we're going to take a closer look at that. So what you see in that folded over bit of strand, actually just one screen up for a second, is you see these little things that look like balls or eggs, right? And you know, that strand could be carbon fiber or it could be a hair fiber. We don't know what it is. So let's scroll down and we'll have a look at the X-ray spectroscopy to see what exactly those little ball shapes and strand shapes are made of. So spectrum 60 is one of the balls that wasn't in that folded part. Spectrum 61 is the crystal. Spectrum 62 is the folded over part of the shaft and maybe one of those little round balls, but we'll see more of those later. So let's scroll down. If we look at spectrum 60, again, we see a whole lot of carbon, a lot of oxygen, some calcium, some sodium, and some chloride, silicon. No signs of nitrogen, no sign of phosphorus. So we know this is not biologic. This is an entirely carbon-based structure. Let's scroll down to spectrum 61. So again, pretty similar story. Carbon, oxygen. For some reason, the square under spectrum 61 accumulated a lot of platinum. But again, platinum could be something that we added to the sample just to protect the sample from being burnt up in the first second or two of exposure to an electron beam. But again, no signs of nitrogen or phosphorus. It's just carbon, oxygen. So we know that there was nothing biologic in spectrum 61 in that square. And then spectrum 62, same story again. Carbon, oxygen, some silicon, some sulfur, some potassium this time, a bit of calcium, but no nitrogen, no phosphorus. So nothing we saw on that photo was of biologic origin. It appears to be a primarily carbon-oxygen structure. So then let's move on to the next file so we can get a closer look at those things that look like little eggs or little round spheres.
Was that in the same file or do we need to switch to another one?
We need to switch to the next file. Okay. And this is all in the Moderna. So we're seeing multiple different shapes showing up in the Moderna vaccine. This one's actually the Pfizer picture. So those little shapes were from Pfizer. So here it looks like there's two completely different types of strands. One strand looks like it could be a hair fiber or a dead insect leg, right? An insect leg would kind of look like that.
But
except it
again,
doesn't have any
if that was a hair or an insect leg, we would see biologics, right? Like we would see that it would contain biological elements.
elements. Yes,
Okay.
it would contain nitrogen or phosphorus.
Okay.
So we ran the spectrum on that thing that looks like an insect leg, nothing biologic, no nitrogen, no phosphorus. And then we have this thing that looks like a fiber with bulbs growing in it. So let's scroll down and we'll get a closer look at these bulb-shaped things. And we took a spectrum analysis of that, the surface of that bulb. So if we keep scrolling down, we'll see that spectrum 4 is again made of carbon and oxygen. Platinum is stuff we added to the sample, and there's a little bit of chloride. So I did a bit of research into what kind of shapes can carbon take in an electron microscopy. So we looked at carbon fibers, and there was something that's relatively recent in technology development, and that's carbon nanospheres. So if anyone looks up pictures of carbon nanospheres on the web, and they look at pictures that have been taken with an electron microscope, the carbon nanosphere looks surprisingly similar to that bulb-shaped object. However, what I haven't been able to find in the literature, the scientific literature, is carbon nanospheres being grown out of carbon nanofibers. And that appears to be what's going on with that strand with bulbs growing along it. I guess it's a carbon fiber that was somehow manipulated that it would start growing carbon nanospheres. And then the question is, what on earth is this doing in an injection intended for use in people?
What are they? What are they for? What were they developed for?
I wasn't able to ascertain what exactly carbon nanospheres are used for, but I would imagine that carbon nanospheres would have some kind of industrial use, the same as carbon fiber, right? Carbon fiber is used to make very strong materials. But again, I can't explain all these different shapes, crystals, chips, strands, bulbs, all being made of carbon, all showing a complete absence of nitrogen and phosphorus, and all being seen within a Moderna sample, right? And this is the most unusual thing, is it appears that that Moderna sample, if it had any mRNA or DNA in it, there's no signs of mRNA or DNA, because mRNA and DNA are both composed of nucleotides, and nucleotides have nitrogen and phosphorus. So that particular Moderna sample from somewhere in Canada has no RNA in it. It has no mRNA. Or if there was mRNA in it, somehow it's disappeared after being left unrefrigerated for anywhere from 1 to 2 months. I think it's probably closer to 2 months.
Mm-hmm. So with that refrigeration process,
Mm-hmm.
and the fact that, perhaps the idea is that what the design of these vaccines and whatever they're delivering, it needs to be kept cold, obviously, or else it destroys. So what do you say to that when it comes to then injecting it into the body? Because once it's injected, I have to assume that it very quickly gets to a body temperature. And so it just seems very, confusing to me about the fact that it needs to stay so cold. And then what happens to it once it warms up? And clearly your images are showing what it looks like after it's been unrefrigerated.
Mm-hmm. So what we need to do is, I need to be able to get a sample of freshly refrigerated of Moderna or Pfizer and get that to the research group that can examine it under the electron microscope. And if we don't see any of these types of structures, then we have proof that, when it's refrigerated, these structures are prevented from assembling. And then after room temperature or body temperature, then these structures are somehow designed to assemble into various different forms, from chips to crystals to fibers to nanospheres, it looks like.
Concerning to you? What do you think?
Well, it's a complete lack of disclosure, right? Did any press release from Pfizer and Moderna say, oh, in addition to the lipid nanospheres and the mRNA, we're also putting in a bunch of carbon-based experiments into the vaccine? That was not said in any press release, any official documentation. I don't think that was even disclosed to governments, that there is some type of carbon-based technology in all these injections. But before we go on, I just wanted to show the couple files of the Pfizer, just to show that there's strange structures growing in the Pfizer samples as well. So if we could pull up the Pfizer files. So this is what we saw in the Pfizer sample, and in a droplet of the Pfizer, you'd see literally thousands of these little squares. So we put it under the electron microscope, and then we took 2 samples for it to examine what elements are in spectrum 32 and I think spectrum, I can't see clearly on my screen, but no, spectrum 36 and spectrum 37. So let's scroll down and find out what elements were there. So if this was a salt crystal, we'd expect to see sodium and chloride. And what do we see? In square 36, it's carbon, oxygen, a little bit of sodium, a little bit of chloride. But again, it's carbon, oxygen, platinum, which was an artifact because we added that to the sample, and a little bit of silicon. That's really unusual. And let's scroll down to spectrum 37. And again, in that particular square, there's only a tiny bit of sodium, no detectable chloride, and it's carbon, oxygen with a very little bit of silicon. So again, it might look like some type of a salt crystal, but it's made up of purely carbon and oxygen and possibly hydrogen. But again, we don't know how much hydrogen's in that sample because the detector can't detect the spectrum for hydrogen.
So again, no biologic material there.
No signs of a biologic material. So let's move on to the next Pfizer file. And similar to the Moderna samples, the Pfizer sample also had fiber-like structures. So we have a fiber-like structure, and this sample was from a Pfizer sample, and spectrum 46 is on the fiber, spectrum 48 is on the fiber, and spectrum 49 and 50 are from the crystal. So let's scroll down there. So 46, which was also on the fiber, is carbon, oxygen, calcium, some magnesium, some silicon, and it has a bit of an unusual element from the lanthanized series, TM. TM is number 69 on the periodic table, called thulium. And again, that's a very unusual element to see. Like, I don't know what thulium is doing inside the Pfizer vaccine, but there is a detectable amount of thulium.
Is that metal? Is that from the metal?
Yeah, it's a metal. It's a type of lanthanide metal, which is it's not something you see every day.
Okay.
So then let's scroll down spectrum 47, and that was on the fiber part that was seen in the Pfizer sample. Spectrum 47, which was another part of the fiber, again, same thing. Carbon, oxygen, a bit of calcium, some sodium chloride, so there's some salt on that part of the fiber. But again, thulium, silicon, the TMs are showing up for some reason. So there's thulium in the Pfizer sample that we didn't see in the Moderna sample. Again, for what?
Is there a chance that that's an additive again, or no, this isn't something normal?
Quite possibly. It's an additive. It's a very unusual additive. I don't know what the biologic purpose.
I just mean an additive in the lab so that the sample doesn't burn under the electron microscope.
Oh, no. It's only platinum, palladium, or gold that would show up as an additive from the lab. Thulium is a very unusual contaminant.
Okay.
Yeah, it's a very unusual. Like, I don't know, I wouldn't. It would be presumptuous for me to say it's a contaminant. It could have been added intentionally to the sample, and when it forms fibers, it gathers the thulium for some reason and incorporates it in the fiber for some reason. So again, this is a lot of questions. What is exactly going on? So let's keep scrolling down. Spectrum 48, I believe, was also from the fiber. So this spectrum 48 didn't have any thulium. It was just carbon, oxygen, magnesium, silicon, and a bit of sodium and calcium. So again, spectrum 49, similar story. It's carbon, oxygen, and then spectrum 50, I believe, is the same story again. Carbon, oxygen, a little bit of silicon, and then keep scrolling down. And I think spectrum 51, can we scroll back up to the top? Has a lot of silicon in it. So spectrum 51 was that little dot, that bubble that didn't seem to have any shape. For some reason, that bubble has a lot of silicon in it, but it doesn't quite look like a computer chip. Spectrum 49 and 50, that was from a similar looking crystal that we found sitting beside the fiber. So again, if we scroll down to spectrum 49 and 50, we see that it's similar to the previous Pfizer crystals we saw in the previous file, that it was made of carbon and oxygen. So we have polymorphic, which is many different forms. They all seem to be made predominantly out of carbon and oxygen. And they're in both the Moderna and Pfizer samples. And they seem to be in fiber forms. In the Moderna sample, the carbon-oxygen structures seem to be taking nanosphere forms and crystalline forms. And in the Pfizer sample, which I believe didn't have quite as long a transport time as the Moderna sample, so it may have been left at room temperature for only about a month, whereas the Moderna might have been at room temperature from 1 to 2 months. The Pfizer ones seem to only be forming fibers and crystals. So again, what are all these things doing? Carbon oxygen can certainly be a sign that there's graphene in it, but how do they make graphene take all these different shapes, right? From spheres to fibers to crystals. This is a technology that I am not aware of with my science knowledge. So the story with these electron microscopy photos is that a few samples came to my personal possession of both Pfizer and Moderna, and unfortunately both these samples had to travel for an extensive period of time in various vehicles. So they've been unrefrigerated for up to 2 months. I don't know exactly how long they've been left unrefrigerated.
And so normally these vaccines are supposed to be kept quite cold, correct?
That's correct. And with the Pfizer and Moderna ones, what's unusual is that the refrigeration temperatures are supposed to be minus 40, in the minus 40s for the Modernas, and I think in the minus 70s for the Pfizer. And that's very, very unusual, because no biologic reaction requires anything more than minus 20 to be completely frozen in time. So, if there's a biologic specimen, a specimen that has biologic activity, you rarely see ever any temperature for refrigeration less than minus 20. Then which brings us to, well, what kind of chemical reactions require extreme cold in order to be halted, right? And so we're looking at free radical reactions, we're looking at polymerization reactions, we're looking at pretty advanced chemistry here, not the type of chemistry you can do in a regular chemistry lab. We're talking advanced chemistry that requires super cold temperatures. So again, that was the first very big warning sign that there is something very wrong with these injections called vaccines for COVID-19. The refrigeration temperature does not make any sense. And that's a huge question here, is, given that we're seeing all these shapes form inside these vaccine samples after they've been left at room temperature for an extended period of time, what on earth are these injections doing inside of people's bodies, right? Because there's plenty of carbon and oxygen available in people's bodies. So if this is some type of technology that can use carbon and oxygen that's in the environment to self-assemble into fiber-like structures, crystal-like structures, bulb-like structures, is this actually going on inside people's bodies after they get the injection? And then does it take a long time? Does it take 2 to 3 months for these structures to gradually form inside the blood? And what are they doing there, and what's the purpose of them being there?